First of all, I'd like to introduce very briefly myself. My name is Isis. I was a fellow. This is my third BioSummit. I was a fellow last year. This year, I was one of the pod leaders for the new fellows. So I have been in all the cycles of the BioSummit. I was a participant, and in the first BioSummit, I participated of some panels too. In the second fellow, now the pod leader and helping to organize. And I know most of the people that are going to talk today, and they are all amazing. And I was just introduced to Jenny, and I'm sure that we will love to see everything that she has to share too. And I will start with Sarah. Sarah is the founder and director of BioBlaze. She is a PhD in plant biotech, and she has been working for years with Open Bio and Citizen Science, and she will be great being a part of this panel. We have Harry. Harry was a fellow too last year, together in the same pod that I was, and he is one of the founders of the Hive Biolab, and he's from Ghana, and she's also involved for several years in Open Bio and Citizen Science, and will have great contributions to tell about his history here. Jenny is a researcher. She's a researcher in UK, in Cambridge. And she is also a co-founder of nonprofit Beneficial Bio. And Scott is also part of the BioSummit since the beginning, right, Scott? He was in all the editions of the BioSummit, and she was already in all roles that you can imagine here, and always helped to organize everything. And she's also involved, of course, in the Open Bio movement, but in Canada. So I think we have a great team here. Sarah will be the moderator, and the rest will help, will share all their experiences with us. So I'm very excited to listen to you guys. Sarah, I'll give the word to you.
Sarah, you're muted.
One second. Okay, thank you, Isis. Yeah, so I'm Sarah Ware, founder of BioBlaze Community Biolab. It's in the Chicago area. And I've been to all 4 BioSummits. So I actually just found a clip from the first BioSummit from the Hello World, and I'm going to actually try to show that in my session later on this afternoon. But it's really funny to watch because I said I'm going to try to start a community lab soon in a makerspace in St. Charles, Illinois, and the next month I did that. Anyway, so yeah, I've also been a fellow last year and organizing things, but it's great to be back, and I wish it was in person, but I'm glad it's not, obviously. We have some great panelists here, and so we already heard some earlier today, or at least I was in the session with Manu when he was talking also some about frugal science, and it was amazing. So we want to kind of continue that discussion now and just hear from some of these great panelists that we have, Harry and Scott and Jenny. And mostly what we are going to be discussing today, we decided won't be like the hardware side of things, because I know that's also an important aspect to be frugal with. But we will be talking mostly about reagents, the reagent side of frugal things in science. And so I want to just start with, I'll start with myself, and then we'll have the panelists introduce themselves briefly, but then we thought it might be fun for all of us to share one of our favorite frugal science tricks, or something that we've figured out that we can substitute just fine. And so I want to see if I can share my slide really quick. I only have one slide for me because it's mostly about the panelists, but let's see if it will work or it won't work. I actually am, one second. One of my favorite, I've been too many things. Okay, one of my current favorite frugal things, substitutes, is a substitute for silicon dioxide. And at Sigma Genesis, 1 kilo of this is $328. And I mean, that's a lot, it's a kilo, but I got mine from sciencekitstore.com for $15 for nearly the equivalent amount. And what we're using this for in my lab and some other labs is to do a really frugal nucleic acid extraction method without columns, without commercial kits. And I mean, this is enough to do like a lifetime's worth of these kind of extractions for $15 using this. So anyway, that was my frugal share. So I'm ready to turn it over to the panelists, and let's see you introduce yourselves briefly and let us know what is one of your most favorite frugal science bits you can share with us. So it doesn't matter. Jenny, you want to go?
Sure. Hi, everybody. My name is Jenny Malloy. I'm a Shuttleworth Fellow at the University of Cambridge, as Issa's very kindly introduced. And so we work on producing frugal enzyme manufacturing protocols. And so one of my favorite tricks that we've been trying recently is producing lactose using milk. So using milk and vinegar to produce whey and sterilizing that to use it to induce the expression of enzymes that normally you'd need to add IPTG. So that drastically cuts the cost of our enzyme expression. And I like it because it kind of exemplifies some of the ways of patterns of thinking that we're trying to embed in our work, which is around use of local resources, use of environmentally friendly reagents as well, and really trying to kind of bring the cost down, but in a way that's accessible for people, and it works, which is always a bonus when you do a frugal lab hack. So that's probably my favorite of the moment, although there are many. Thanks, Sarah.
Yeah, great. That's a good point. They don't all work. That's true. But the ones that do, they're great. Yeah.
And
go ahead.
my favorite is actually one of many is trehalose. So it's a disaccharide sugar, and a lot of community labs, they just can't afford to run a -80 freezer. Expensive, the electricity is expensive, and my -80 freezer developed a fault a couple months ago, so still trying to figure out how to fix that. But this trehalose, you can actually mix it together with your bacteria. So if you're working with plasmids and E. coli, of course, typically you freeze them at -80 to store them. But with trehalose, you can mix that in and dry them down, and you can end up with a nice stable for at least 5 months or so. And so you can get trehalose on Amazon dirt cheap, maybe about $46 or something, well, Canadian for about a kilo. Give it a go.
Yes, so for me, my name is Akligoh, and like I said, I'm one of the founders of the Hive Biolab and a researcher with the Open Bioeconomy Lab, where we are exploiting the open enzyme collection for producing DNA polymerases locally and using that to set telemolecular assays for identifying ACIs in Ghana. And my favorite parts with regards to frugal kits, lab tricks, is the squeeze and freeze technique for cleaning up DNA. I think this is very frugal to the latter in that you basically are just making use of your fridge and just a piece to clean up your DNA, which I think is very sustainable in very disadvantaged backgrounds where access to reagents is usually a challenge. So I think this, for me, is one thing that really got me, especially where you do not need to use any kits in DNA cleanups and all that. Thanks.
Yeah, thanks, guys. I think it would be really cool. Hopefully there's a document where you guys can go and take notes and add your own frugal ideas, because I actually didn't know any of these other three, and I am excited to try them out. And so please, if you have some of these, put them in the notes and we can all be more frugal.
The power of
So
community.
exactly,
Yeah.
but we have to have a way to share it, and we have to have a way to access all of our collective information and our knowledge. Okay, so you haven't mentioned this before, but it's something that I know is of interest to everybody in the group and will also be of interest to the community regarding frugal science. And that is, could we get a report on the status of the age of open source DNA and open source enzymes? Like, where are we at right now with those things? Harry?
Thank you very much. So I would say my question and response to the question will come from an African perspective. And I would say that currently we are in the budding stage of harnessing the potentials of open source DNA and generally the open source enzymes. And this revolution, as we have seen it, largely facilitated by the completion of several sequencing projects, largely one which we all know as the Human Genome Project. However, the utilization of this open source DNA and its resulting enzymes collection has been underutilized on the continent. So I would say with regards to status, that is what it is. But on a global scale, I think quite a lot has happened. And I'm sure my other panelists would share to that. Thank you.
Yeah, thank you for your perspective. And Jenny, would you like to go ahead and share what you got on all of this?
Sure. Yeah. Well, I think in terms of so Scott was the one that kind of proposed the age of open source DNA and what that means for the world, which we're here to discuss. I think for me, I think we're at the beginning of that. Lots of work has gone on before, but we're really at a golden age where we've both got a bunch of really important technologies coming off patent. Their 20 years has expired. They're now free to the world. They're public domain. And we're kind of, that 20-year window is about to sweep through the early 2000s and the genomics era, which is very exciting. And on top of that, you've got communities like this one, which are pushing open source kind of DNA and open source designs and open source science out into the world. And so I think we've just got an abundance of material right now. And I feel like we're kind of, we're a bit like open source software kind of in the 90s, where there were lots of projects happening and there was stuff coming out and there was a great deal of excitement and anticipation. And I think how the next few years are going to be interesting as to how we harness that and bring it forward. I think we still have work to do around, the pragmatics of getting that DNA out there. We have a lot of it, but really making it used and useful and getting to people's benches is going to be kind of, I feel like the status is we're not quite there yet. And we have a lot of organizing and hard work to do. But yeah, I mean, we're definitely at the beginning. I think we're well into the start of this open DNA age or era that that Scott kind of posed, and yeah, I'm hopeful. I wondered if Sarah, we could just share those 2 slides on the open source enzyme collection, just to show an example of one of the initiatives that several of us on the panel have been involved in, to just give an indication. So I was quite surprised this didn't already exist, but as I mentioned, we have, there's so many enzymes, so many DNA parts that are open source effectively in terms of their patentable status. And so I just wanted to raise awareness of this one particular collection among many, the Open Enzyme collection. I don't know if it's possible to put it in present mode because it's quite small on the screen there. I think it's view and then present if possible, or that one, yes. Great. And so just to describe what the Open Enzyme collection is, so we have 2 slides back. A whole collection of different parts. So we have 10 tags, 31 reporters, 5 cleavage sites, over 60 enzyme sequences that are all now real. There's physical DNA that's being distributed under an open MTA. And if you click to the next slide, you can actually combine these into nearly 200, well, we have nearly 200 interchangeable elements that you can recombine. We also have some expression cassettes that are being synthesized right now and should be available quite soon for really common lab enzymes. And so if you click to the third slide, just to say that, you know, as I've just mentioned, the age of open source DNA is definitely upon us, and you can order these open source enzymes literally free, as well as being free in terms of the public domain. And so Stanford FreeGenes have these collections. You can see at $0, they will ship them to you. So if you would like any of the open enzyme collections, they're currently in 2 parts, and Open Reporters as a separate collection. But we're aiming to do a kind of combined one at some point. And at the end of the month, I'm just going to give a quick kind of heads up. We're having a research and diagnostics collection ready for distribution, which contains many enzymes that are useful for molecular diagnostics. So watch this space for that. Also will be available at the same place. And so this is one of the projects we've been working on that kind of gives me a lot of optimism about the future of this open source DNA world, but we've got a long way to go.
Yeah, that's great. Thank you.
Sarah, if you can advance to the next slide.
Sure.
So tomorrow I'm going to be talking about the Open Yeast Collection. And this has been a project that I've been developing ever since I first heard about FreeGenes and the BioBlaze Foundations with the Open MTA. So I'll be giving you a lot more details about that tomorrow in my workshop. But I just want to bring it up. It's currently not yet synthesized. I pushed FreeGenes to make sure that Jenny's work got synthesized first in terms of priority, because that's a foundational tool. And in fact, within Jenny's Open Enzyme Collection, there are the enzymes that you need to assemble that collection. So if you wanted to make those 200 different diverse things, you can produce some of those enzymes to do that. Jenny broached the topic of open source software, and for the longest time, I grew up watching this movement start. In the very beginning, it was a system where, just a bunch of people got together and they thought, this is cool, let's play, let's do this, let's explore. And they released it out into the public, open domain, public source. And it evolved. And if you think about all of what we're using now, the internet, if open source software didn't exist, if it was all commercial software, think what the internet would be like today. Would we have something like Zoom? Would we be able to have a free account on social medias, things like that? Would we be able to put up our own websites that tout our own, community labs, things like that? So I draw these parallels with the open source software. It's really a small community of folks as it started, and people were sharing, people were excited, they were playing, and through that evolution, that interchange between everybody, new ideas come in, new people come in with new ideas, and you start to ferment this tremendous possibility. And so things like the Open MTA is incredibly powerful for creating this distribution. And so, yeah, as Jenny said, we are just on the cusp of where we want to go. And, I'm thinking that if the Open Enzyme Collection was fully printed and distributed before the pandemic, where would we be? And Sarah can actually talk about her work with the Just One Giant Lab Open COVID-19 initiative. And we're doing a talk there. So those diagnostic tools use enzymes in this kit. And so if these were distributed far and wide already before the start of the pandemic, where would we be? Sarah?
Yeah, I think that's a really interesting question, because right now we are in it and we are kind of really, really scrambling you know, we've been talking about this for a while, and there's been a ton of work that's gone into it. Jenny, you didn't give yourself credit, and all of you, but there's been a ton of work going into this that people don't see. And but yeah, what if we had these already ready to go? That means, like, right now we're working towards that while we need it now. But we're going to get there. But yeah, I mean, that's a really big what if, huh? What would be different with our testing? We're kind of almost where we want to take some questions. I know it goes so fast, but from the people, you know, out listening. But we had another couple questions that maybe one of you could tackle quickly before we start taking questions from everyone else. One is that, what is one major accomplishment that you hope will emerge from an open DNA collection and an open enzyme collection within the next 5 years? Who would like to field that one? Jenny?
I'm willing to pass on that one. I think I know the upcoming prompt, and I'd rather answer that one. So I'm going to leave Scott or Harry to pick it up.
Yeah, sure. You know, I think, you know, if we use the history of open source software, so we are on the early stages. So I think a part of it is really about making the connections and making the distributions, getting these things out there. And I think what drives innovation is curiosity and play. You know, I think play is just so important. We live, you know, I kind of talk about we're all, we all operate within a context, and everybody's context is different. We often limit ourselves because of what resources we have access to. So if people have these things out there, some people may pick it up and play with it in one way, another person may pick it up in another way. And then if we collaborate to contribute, so for the Open Enzyme Collection, Jenny can talk about Reclone, about sharing resources and protocols on how to actually distribute, produce these enzymes. I think these things are super powerful. So I see in the first 5 years, a period of distribution of quick innovation. There'll probably be overlap. There'll be all sorts of things. And it will coalesce into a great many things. And I think it's really going to be an exciting time. You know, DIY Bio has been a lot about hardware. You know, when Cafe Garvel developed his centrifuge, his Dremelfuge, way back, when was that, 2009 or so? Somebody here might probably know. It's powerful, but it's been a history of hardware because that's what people could access. But now we're putting out these DNA. How can we get that? Keamy talked about Bacillus subtilis. Spores are a great way to send DNA around the world. So there's a lot of potential, a lot of opportunities.
FreeGenes is amazing. And like, I even tell people at the universities where I teach about it, I'm like, you don't have to be a DIYer to use FreeGenes, and they just insist on buying them. So I don't know, but so Jenny, we want to, oh, go ahead, Scott.
Oh, I was just going to say it's the kiddification of science, and you know, I call it the biomedical industrial research complex. All of these companies that are feeding off of public grant funds, largely, they're making a ton of money. When you get a tiny little tube of Taq polymerase that you could have produced for maybe 3 times the cost of that tube, and it's enough Taq for the rest of your lab's life.
Yeah, you know. Okay, Jenny, you want to weigh in on the last thing? And then Harry, if you have anything also after that. But so what do we need to do most importantly to maximize our democratization and distribution of this open DNA and open enzymes and reagents?
I'll hand to Harry first.
Thank you very much, Jenny, and to Sarah. I think largely what we are seeing is would create a more equitable access to tools for researchers, especially those in disadvantaged backgrounds who do not have the tools to do research. That is what I see happening largely, and a lot of capacity building happening for these scientists in underserved backgrounds to get into this. Grounds to have and use the tools that are coming into the public domain, especially with open source DNA and all that. And I think other groups are leading the efforts, which for me, it's very interesting for me, especially from the background which we come. So these are some of the things I think are going to be happening in the long term, and things generally that needs to come in play to put everybody on an equal level playing ground.
Yeah, I mean, I agree with everything Harry says. And I think one of the things that we really want to see with the collections, and I know Scott and others who've put effort into collections want to see this too, is like you want that collection to catalyze a cycle whereby people use it, that communities of researchers use the tools, contribute back into the collections, and this kind of enables the next generation. I think that's that's hard. It requires not only the technology, which we have, but also the sort of social organizing and the good governance. I think one of the things I like about the BioSummit community is that's what community biology is all about. And so I think it's one of the reasons this community is key in sort of operationalizing the age of open source DNA. We have the DNA, and now we need to actually use it and see this kind of virtuous cycle being created. And getting that feedback is much harder than in open source software, like getting the kind of the people innovating on the collections and contributing them back. But I think it's critical to kind of move this forward and have people doing stuff with the collections that, you know, was unpredictable. I mean, it's a win for me if in 5 years down the line, somebody is making something with the Open Enzyme collection and contributing tools back that I could never even have imagined, and that they could never have done without those tools. Because I think that really would be proof that we're changing the trajectory, not only of what an individual scientist or a team can accomplish, but also kind of, you know, moving it beyond the small group of people that catalyze these things into a community that takes it to places that we could never have dreamed of. And so that's what I'd like to see. And I think that's, you know, some of those things that we need to happen in the next few years to make this, to drive this forward.
And Jenny, do you want to comment if people have ideas about genes? You have a resource to capture that information?
Yeah, tell us. If you go to Open Bioeconomy website, you can request an enzyme, pick your favorite. You sort of have to justify it a little bit in terms of its usefulness to the research community. But yeah, head on over and add something to our pipeline.
Yeah. Just vet it, or let us know that you haven't vetted for patents, because the more you work you do, the easier it is for us.
So do we have any time for questions, hopefully, or
we squeeze one?
30 seconds.
Okay, sorry, I gabbed too much.
We have 30 seconds for a wrap up. If anyone would like to join from the panelists.
Put your hand up if you want.
Oh, that's a good one too. All right.
Oh, thank you.
Yeah. Thank you, everybody. That was amazing. I think that everybody here learned a lot with all the contributions. I learned a lot.
I've been learning so much in this chat, can I just say? Like, I need all of the links.
Yeah. Well, you can actually save the chat, so.
Excellent.
Yes, and the chats will be saved. And also, please, everybody that is attending, don't forget to check the panel doc and the main doc, because you can keep contributing and exchanging there.
Thanks, Isis, for guiding us through it all. And Sarah for being our chair.
Thank you.
Thank you all, guys.
Thanks, everyone.
Bye.
Bye.
Bye.